My name is Victor Abba, the Laboratory Manager for the Center for DNA Forensics and Criminal Investigation, CETFOC. I am a biochemist who majors in health studies in molecular biology. Okay, the reason for the experiment, which we explore biodiversity, is to find the link or the bridge between field ecology and molecular biology. That way we explore microorganisms in their natural habitats before we collect samples and then transfer them into the laboratory where we explore their DNA diversity, as well as how they fit into solving problems in medicine, pharmacy, and other molecular applications. All right, so our field trip on biodiversity, where we would explore different environments and collect samples. These are our tools we'll be working with. We have sample collection bags where we'll put different samples we're collecting, solid samples, microorganisms related. We have tapes for labeling. We have other tapes where we'll use to create our 2x2 feet or 1x1 feet in the areas we're collecting samples. We have tubes for water samples. We have measuring tapes for our environment. We have cutters, scissors. We have alcohol for disinfecting. We have our flashlights because we're going into the Ugwuomu Forest, and we're going to need flashlights to see things in their natural habitat. We have a mobile digital microscope in case we need to verify microorganisms. We have our magnifier, also a digital magnifier. We have a power source for this digital equipment. We also have collection tools that we would use if we want to prepare slides in the field. We have our tweezers, we have droppers to pick up water. We have our labeling equipment, our pens, our documentation book. We have slides, we have swabs to collect samples. We have cutters for hard samples like tree barks, wood samples. And then we have our gloves for protection, also to collect decaying grasses and leaves on the floor and samples from the earth. We also have our mask, which we'll use to protect ourselves from spores that will try to break out. We also have our external protection gear like our boots that are down there on the floor. Those would help us protect our feet because the forest here could be surprising, so we need as much protection as possible. So as you get ready to take this journey with us, you're going to see just exactly how biodiversity moves from the earth to a petri dish. Join us as we move ahead. Now we're going to load up our gear so that we can move into the forest and begin to explore biodiversity. So all our gears will go into our bags. Ensure you have all your tools because you don't want to be in the forest and begin to remember that you forgot some equipment that you would need. Also, it's important that you have some sort of GPS so that you know exactly where location you're working with, and you also don't get lost while you're in the forest. So okay, so we are all set and ready to go into our forest. So we're currently at the edge of Ugwuomu Forest, and we're about to dive in where we're going to be exploring biodiversity in its most natural form. So this is one of the least interrupted sections of this forest by humans. You can look at the bamboo trees, they're uncut at the stem. You cannot see cut bamboos. I mean, that's one way we get to know places that are uninterrupted. If it was interrupted, you will see multiple cuts at the bottom of the bamboos. This is the beginning of our swamp area, and this is basically an uninterrupted habitat that we're trying to explore and bring as much samples as we can back to our labs so that we can research and do a whole lot more work around the forest. So we can see that the untouched bamboo trees go over 20 meters tall into the sky, and that is why many of them begin to bend, because they're so tall. And unlike the Chinese bamboos, which are very strong and can go taller, most of these ones later bend sideways. First of all, our experience shows us just how diverse life is in the forest. You take small samples and you see a large amount of biodiversity of different microorganisms. You get to see how much precaution you need to take as a scientist in the field, how to collect those samples, how to preserve and transfer them into the lab for further experiment. You also get to see how much diversity there are from the leaves, the plants, the soil, and different organisms in their natural habitat. And the reason we're doing that in the forest is because it is the least interrupted area of the environment where we have non-external factors playing in that particular habitat. Almost everything here is uninterrupted by humans and it's untouched. So when we log in, you can see the swamps. There are no footpaths to the swamps. So we're coming to a part of the forest that is least interrupted. So this is one of the least interrupted sections of this forest by humans. You can look at the bamboo trees, they're uncut at the stem. You cannot see cut bamboos. I mean, that's one way we get to know places that are uninterrupted. If it was interrupted, you will see multiple cuts at the bottom of the bamboos. This is the beginning of our swamp area, and this is basically an uninterrupted habitat that we're trying to explore and bring as much samples as we can back to our labs so that we can research and do a whole lot more work around the forest. So we can see that the untouched bamboo trees go over 20 meters tall into the sky, and that is why many of them begin to bend, because they're so tall. And unlike the Chinese bamboos, which are very strong and can go taller, most of these ones later bend sideways. Imagine you can pass our place. Because I'm not sure I can. Wait first, let me take our longitude and latitude locator. 759, this one is 736. So right now we want to take the specific location of where we are. So our GPS puts us at longitude 633.45 north and 736.16 east. We're at an elevation of about 390 feet above sea level. So we have a swamp where we want to collect some water samples that we will take back to the lab and process. We can clean the bag later. All right. So first off, we want to collect some water samples, and my assistant Emmanuel would help us with those samples. You also would like to ensure that you label the exact location and source of your sample so that you do not mix them up. We have our first water sample, which we've designated W for water 1, and we're also going to inscribe our location so that we know exactly what body of water we're talking about. So, having labeled our sample, now we can go ahead to collect our water sample. Please, if you see any stone with algae, let me know. So at this point, if you come with me, you can see on our bamboo trees, you have microorganisms living on the surface all along these joint areas. And if you look at this particular one, you can see gatherings just around this area. Now we don't know what they are, but we can take samples, go back to the lab and see what kind of microlife we can find around this particular bamboo. So to take samples, we have to also label our sample collection. So we'll call this B1 because all of 1 are designated to the samples collected at our first location, which we've already gotten the geolocation for this particular samples. Can I get a cutter? So often people wonder, what is the importance of what we do? Biodiversity is the first step for us to finding solutions to a lot of pharmaceutical products, to find sequencing, and biodiversity environments are environments where we can get in touch with organisms that improve the well-being of life and that can also provide a resistance to diseases and all of that. So a lot of this is very important to what we do in the lab to find solutions. Let me hold this. So what we need is just a little cutout from our samples, and we want them to just fall into our bags. There you have it. Now we can take a little bit of the life forms that would also live around this area. So, and there you have it. Now, even though it might look so small to the naked eye, you have no idea what you will find under the microscope for just this particular sample we are collecting. It's very important that we get some soil samples so that we can see the variety of microorganisms that we have at this particular spot. So of course we labeled our sample container, and then now I could simply get a spatula out of my bag so I can fetch the particular sample. So right here we have a little bit of sun that passes through. You can see the soil is very moist and damp. This kind of soil are very rich in decomposers, so we just want to be able to cut out of it, take some of these samples and put in our container. Now we want something that's very rich. That we can take back to the lab and see what kind of microorganisms we are going to find. So we have some samples from this area. We want to go deeper into the forest and see what we're going to find. So, are you ready? Let me know when you're ready. Kind of like stones or rock body underneath the ground. As you can see, we have an earthworm that is scavenging, looking for food. To this side, just where my hand is, we have a whole ecosystem of microorganisms. We have moths, and I'm sure there's so many organisms we cannot see with the naked eye. So it's only good if we take some samples. So I will just try to cut off and then put in my sample bottle. Try to get as much of this. So we're also going to take some and put in a petri dish so we can take to the lab and explore everything that lives right here. Yeah. Huh? Okay. No. Okay, so we're in an area of the forest where it seems there are a rich array of flowers growing here. I mean, it's incredible. These are flowers we see in savanna regions, but somehow they are thriving in the forest. We can see we have pink flower, we have the sunflower right here, the Nigerian indigenous sunflower. We can see rich insects because this is a very good area for pollination to occur. There's another unique flower just over there, also a little bit pink. So if we come right here, we know this is something that is very wild, as we can see. So along this area, we have a rich vegetation of flowery plants that would have a vast diversity of insects. I mean, that's the only way that we grow so well, because pollination is happening in this particular region. You can also snap it. There's no need to kill it. It's actually against science to take it off like that. So you can snap and identify using the pictures. So even while we explore the plant life around this area, unlike other areas, there is something that is peculiar with all the plants. You can see all the leaves are being eaten off for the different plants. We have this that has a very large leaf, also eaten. We have this with very small leaves. suggesting some type of insect or probably disease that is attacking the plants in this region, most likely an insect, because all the different plants have this one thing in common, that the leaves are being attacked in this specific area. So right here we have a decomposing branch from a tree, and we want to see what kind of life is on it. Okay, so we notice that at the heart of this forest are swamps that are connected on every side. Right now we're deep within the forest, and we just came upon an opening to the swamp where we're seeing all kinds of life forms, both in the water and then right here at the edge of the swamp. So we're still going to take one more water sample from this side and see how it connects to the samples we collected in the other location. So we're taking a sample from a location where we have a lot of leaves in the water. So here we have our sample, and even with your eyes you can see a live creature swimming within the water. All of this would be explored back at our lab. So every research and every lab work will be done at DNA Learning Center Nigeria, where we're exploring solutions in science to help Nigeria to move forward. Thank you. Here we've located some mushrooms. We can see it on this branch. So we're just going to chop it off. This is one particular sample we've been looking for all day to find, so we can know what kind of mushrooms naturally occur in this particular habitat. Mushroom is something that is very important in this particular region of Nigeria, so it's always good to know what grows naturally in this kind of forest. According to the locals who live in this area, I've described this as a forest where it was called the evil forest back in the day. It was locally known as the place where twins were being dumped because it was seen as kind of like sacrilege to give birth to two children decades ago, hundreds of years ago here in this particular region. So it's a forest with a history, rich in cultural significance to its people, and then we can see that science gives us the leverage to venture into what people termed as dangerous back in the day, to explore solutions in Africa. Right? Okay. So this episode of Biodiversity, from the earth to the petri dish in Ugwuomu Forest, has been brought to you by myself, Victor Abba, and my colleague,
Emmanuel.
So we'll be taking you through all our science discovery in biodiversity in our series. So join us as we journey through this rich cultural forest in Ugwuomu Nike. Alright, good afternoon. So we're here to talk about biodiversity. What does biodiversity mean? We can break it down to be biological diversity, which simply refers to the variability of life. How diverse is biological life? So, why is biodiversity important? It's very important because it feeds into almost everything we do in science, specifically molecular biology. We have its industrial applications, its medical applications, its environmental applications, and so on and so forth. Now, this is very important because in science, where we're trying to break forth and also preserve our environment, biodiversity gives us the information to do so. When it comes to getting medical solutions, vaccines, pharmaceutical products, biodiversity is basically the hub from which we can find biological solutions to biological problems. Now, what are the types of biodiversity that is out there? we have our genetic biodiversity, which is the variation of genetic information or genetic life. We also have that of the the ecosystem biodiversity, that is the diversity of different and various forms of life in an ecosystem. However, we're going to be focusing on genetics and that biodiversity because that is what is important to us in the lab. So our environment where we visited today was Ugwuomu Forest, and we have samples we brought back. And if you stay with me, we're going to be exploring the life form from the different samples that we collected. So here are samples from Ugwuomu Forest here in Enugu State, and we're going to be using different microscopes to help us explore life at its level. We have our digital magnifier, which help us to check just some samples in their whole state. We also have a digital microscope with a 1 to 1000 magnification, and then we have our advanced compound microscope that would also help us to see slides of our samples collected from the swamp. So what I'm going to do is take all of these labeled samples one after the other. We're going to put them in a petri dish and we're going to see what is going on under the microscope. So first off, we had some samples that we took off bamboo trees where we saw some life forms. So we're just going to take the back of the bamboo tree, put it in our petri dish, and try to view what life form could be on the back of the bamboo tree. Can you see it? So we do have a termite that lives in bamboos on our bamboo back. So it's kind of shy of light, so it's in hiding. Right now we can see it moving, and because of the light of our microscope, it keeps going into hiding. So as part of the bamboo, we can see the bamboo termites. We can also see some moss on the bamboo, some green algae together with the green back of the bamboo tree. So these are samples taken off the bamboo, and we would make our time to identify the particular species of termites that we found right here on this bamboo. These creatures are very sensitive to light, and as a result, they are avoiding all the lights on our microscope. So as long as I move it, the lights, it's looking for somewhere to hide. And that is why it's in constant motion. You can see the detail of the bamboo bark from the outer skin to the inside in brown. Okay, so we do have various samples. We've seen one of our samples. So a lot of what we're exploring today are samples of decaying or dead matter, and we want to see organisms that would most likely we would see in this habitat. So right here we have algae that we got from rocks close to a water spot. We want to see what life on a very microscopic level looks like for organisms within this habitat. So we're looking at different parts of our algae. So while to the naked eye, everything on the algae is green. Right here we can see some of the plants are actually red, orange, or purple in color. We would see even more of those when we adjust it to see The algae that looks like pure spikes actually are plants. And we can see leaves, like structure, other colored structure, and then decaying matter at this particular point. So this is what life looks like for an algae under a microscope. Now, right now we may not see a lot of light and a lot of activity from microorganisms, so we're going to do further investigation, further experiments to find out what lives in this particular habitat. Right here is what we thought was a dead piece of wood we just picked in the forest, so we want to see what's happening on this piece of wood. So right now we have the wood, and we're going to use a digital magnifier to look at it up close. So this is what it looks like up close. I can get ready. So this is what it looks like up close. We can see that there are plants growing on this supposed dead piece of wood all around it. Okay, so this is what we consider to be a dead piece of wood. We can see how much green life is embedded in this dry, dead piece of wood. As long as there is moisture around it, all of this green life would occur on this piece of wood. We can see a lot of it under the magnifier. And this is very important, and you wonder why. This is important for recycling resources into the atmosphere. This is very important for fixing nitrogen into the soil and also creating oxygen, which we need for a healthy environment for human survival. Ready? Okay, so here we have a mushroom plant growing also on this dead piece of wood, and we can see the life that surrounds it. Just around, this is what it looks like. Right here there's a living organism, if you can see it. It's translucent, you can see it moving. So let's see if we, now most of it is hiding because of the light from our microscope. So of course we cannot see this with the naked eye, but it's right here. All of these are responsible, we can see it now, moving. These are very important microorganisms for breaking down, decaying, decomposing biological materials to the earth that help us to recycle energy. Helps. Okay. So all of these organisms coexist even as it's living within the wood. It provides resources that even the mushroom is going to benefit from to maintain its own growth. So we can see other microscopic growth with another organism right here behind. It's living behind this transparent growths. So most of these organisms would come out if the light on the microscope weren't there. So even on this dead piece of wood, there is a whole lot of life going on within it. And of course, we're going to have to identify each and every one of these organisms so we can talk about them even more. Today has been exciting in Ugwuomu Forest, and we've barely scratched the surface. We've been able to come out with samples, taking it from the earth into the petri dish. Now, what next, you might ask? Is this the end of it? No, this is not even the beginning. This is where samples can be taken for DNA barcoding, samples can be taken for DNA sequences, samples can be analyzed, and samples can be experimented, like just go through all of the samples to find out its usefulness and its benefits in groundbreaking science. Now, this is where we can see results that could influence agriculture, influence that could influence medicine, and could give potential solutions to even our environment. With forest life ever decreasing, work like this goes a long way to help us in preserving our environment, keeping it healthy, climate change, kind of like putting a pause on climate change. So at DNALC Nigeria, we explore science solutions to scientific problems, and we've been exploring biodiversity. From me and my team, I say thank you. Have a great time.